Hi, good afternoon, and thank you for attending today's Congressional Biomedical Research Caucus. My name's Lynn Markey, and I'm the director of the Coalition for Life Sciences, and we're always happy to host these events for you. Today, it is the last congressional briefing that we will have in the 2014 season, so I would like to take this opportunity to thank all of our co-chairs for everything they have done for the caucus throughout the year. It is because of them that we are able to bring these briefings to you. And those co-chairs are Representative Steve Stivers from Ohio, Representative Charlie Dent from Pennsylvania, Representative Jackie Spear from California, and Representative Rush Holt from New Jersey. And before I introduce today's speaker, I'd like to he's not here yet, but I would like to say thank you to Representative Rush Holt for all that he's done. He is leaving Congress at the end of this session, and he has been a co-chair of the caucus since 2003, and he will be greatly missed. So we thank him for all he's done for the caucus and for science. So today, I am pleased to introduce Dr. Carol Barnes. Dr. Barnes is a Regents Professor in the Department of Psychology, Neurology, and Neuroscience, the Evelyn F. McKnight Endowed Chair for Learning and Memory in, I'm going to turn my page, hold on, Aging. She is the director of the Evelyn McKnight Brain Institute, the director of the ARL Division of Neural Systems, Memory and Aging, and associate director of the Bio5 Institute at the University of Arizona in Tucson. I would hate to put that all on a business card. It's quite the title. Dr. Barnes is also past president of the Society for Neuroscience, an elected fellow of the American Association for the Advancement of Science, and an elected foreign member of the Royal Norwegian Society of Sciences and Letters. She recently received the 2013 Society for Neuroscience Jared Prize in Neuroscience, the highest honor conferred by the Society, and the 2014 American Psychology Association Distinguished Scientific Contribution Award in recognition of her groundbreaking work to uncover the changes that take place in the aging brain. Dr. Barnes earned her BA in psychology from the University of California at Riverside and her MA and PhD from Carleton University in Ottawa, Canada. Today we will hear about the central goal of Dr. Barnes' research program, which is to understand how the brain changes during the aging process and what the functional consequences of these changes are on information processing and memory. It is my pleasure to welcome Dr. Barnes.
Thanks so much for the invitation, Lynn, and a kind introduction. Let's see if I can work everything here. Yes, I can. So if I start to fade out or walk away from the microphone, somebody please remind me to walk back. I'm pleased to be here with you today to tell you about some of the exciting research going on in the field of neuroscience. I was president, as Lynn said, of the Society for Neuroscience in 2005, and the Society for Neuroscience is among those that participate in the Coalition for the Life Sciences. And I'll talk to you today about what each of us individually can do to do our part to ensure a successful mental lifespan. One advantage to having been interested in aging and memory for 4 decades now, actually since I was in graduate school, is that I can clearly see the remarkable progress that we've made over these years. And this deeper understanding of the brain and aging, of course, has been enabled by continued support from Congress for both basic and translational research through agencies such as the NSF and NIH. Naturally, each of us has to set our own goals for successful aging, and one of mine is to be able to continue my sunrise walks up the mountain and waterfall behind my house, if possible, at least until I'm 80 or so. And this may not be completely realistic, but it may be. But one thing is clear, is that to continue to enjoy the Sonoran Desert as I do, I'm going to need to keep my brain healthy. So what is a healthy brain? When you're 40, when you're 50, when you're 80, when you're 90? When does the normal variability in brain function cross the line and become a medical concern? This is sort of the kinds of questions that I'm trying to ask. So today, I hope that I'll provide some clarity on what normal brain aging is, or what's happening to most of our brains as we age, and what this means for memory across our lifespans. So being interested in aging and memory as long as I have, as I told you, has allowed me to see that we've really and truly come a long way in separating and identifying myths from facts about the aging process. And to illustrate this, what I'd like to do today is take you through what I believe to be transformative advances over the past half century in understanding the impact that the brain has on cognition. In addition to this, I have another message. In addition to discovering realistic goals for maintaining our own unique, successful aging trajectory, another important point in my talk is that aging doesn't happen just to those of us who can study it. It happens across the animal kingdom. And basically, other animals also have brain and memory changes as they reach the maximum of their respective species lifespans. So interestingly, because of this, we can learn a great deal about the fundamental principles of brain aging and cognition by studying animals who show these changes clearly. Our research has shown that similar age-related memory changes happen over very different timescales in animals with very different lifespans. So you might ask, first, why is the transition to old in humans thought to be 65 years of age? So let's start there. Well, one answer is that age 65 was statistically determined in 1935 by actuaries as the age of typical retirement of individuals at that time when they were setting up the Social Security system. So just to point this out, in the 1930s and 40s, of course, the average life expectancy at birth was about 62 years of age. Well, today it's 80 in the United States. For males, it's 77, females, 82. But today, 65 has remained the conceptual transition point into old age and is used to define sort of a young-old age groups in most studies of brain aging today. Now, again, we've come a long way over the past 50 years in separating assumptions or myths about the aging brain from actual knowledge about how memory traces are laid down in the connections between cells in the brain and how these memory circuits are changed over the lifespan. And while there's a lot of research being done on brain diseases, which is also supported by Congress. One reason that I study brain aging in the absence of disease is because I believe that to tackle brain disease meaningfully, we have to understand how the brain works normally better than we currently do. In other words, you can't hope to fix it if you don't know what it is you're trying to fix. Okay. And so we've come a long way in beginning to understand the details of how the brain stores information or what the biological basis of memory is. And so let's go to my first, my list of myths on this side and what we think now. So the first of these myths is about what happens to cognition during aging. Well, no one now believes that somewhere around age 65, senility sets in, okay? The most common forms of dementia, as you may know, are Alzheimer's disease, which has a specific pattern of brain pathology, or vascular dementia. Alzheimer's disease is very gradual in onset. Vascular dementia, on the other hand, is caused by one or more strokes and is typically more abrupt in onset. So even 50 years ago, the prevailing view of old age was that if you lived long enough, you would eventually get a dementing disease. But today, we know that becoming senescent or living a very long life does not result in senility. That is, developing a dementing illness. So that not all of us are going to have Alzheimer's disease or vascular dementia at the ends of our life. So what we do know today is that Alzheimer's disease, or AD, is not inevitable. In fact, a study that looked, it's the best study so far that's been done. It looked across the United States in a very systematic way of those people over 70 years old, and they found that only 14% have dementia, age 71 and older. Now, 14% is a huge number. It's a horrible number. But this means that 86% of us at age 71 or over are not demented. Now, these are some of the numbers from the study that I just cited. The prevalence of dementia in those over 70, of course, differs depending on the age group that you look at. So between 71 and 79 years, the prevalence is as low as 5% in that age group. But you go up to age 90, it goes up to 37%. But most of us now believe that even if one is reasonably free of disease, there are lifespan changes in memory. At the very least, these changes are annoying, as some of you in the room might have experienced. So while 86% of those over 71 do not have dementia, we do show memory changes with age compared to our individual memories when we were younger. Now, some of the memory problems that are observed during aging implicate dysfunction of brain systems in the temporal lobe regions of the brain. One such temporal lobe structure, which you may have heard about before, is the hippocampus. One reason for the focus on the hippocampus is that it's thought to be a main gateway for lasting memories in the brain. And we know that, that changes in this brain structure are an important contributor to age-related memory loss. But this is a picture of a hippocampus of a rainbow transgenic mass where different colors, fluorescent proteins, can be labeled in subsets of cells in the hippocampus. Now, we know that the hippocampus is critical to lasting memories because if the hippocampus is damaged in humans or in other animals, memory is severely impaired. And one particular form of memory that the hippocampus is involved in is spatial memory, or the kind of memory that allows us to find our way around our environment. So an example of how we know that the hippocampus is critical for navigation and spatial memory is a positron emission tomography study, that is a PET study, that allows you to measure brain glucose activity when the person is in the scanner. And Eleanor McGuire and her colleagues in London had taxi drivers recall a route that they would take if they were told to pick somebody up at a specific place, say in London, say Grosvenor Square, and then to get them to a spot, drop them off somewhere else, say Elephant and Castle. And when the taxi drivers recalled these routes when they were in the scanner, the right hippocampus, which you see on this left side, at 3 different levels of the brain levels, the right hippocampal activation showed strong positive correlation with them remembering these routes. This is an average of 11 taxi drivers. So these are the kinds of data that support the idea that the hippocampus is fully engaged in this type of spatial navigation. Okay, so it is involved in spatial memory in humans. What do we know about this kind of memory in aging? Well, in fact, it's been experimentally shown that this kind of spatial memory depends on good hippocampal function, and it declines during aging in all species that have been studied. That is, in humans, old monkeys, old dogs, old rats, old mice, all have defective spatial memory. So a summary then of the current thinking about the first point in my myth list concerning how aging results in senility is that while dementing disease is not inevitable, some memory change, unfortunately, is, and just as the example I gave you for spatial memory. And this happens in all mammals that have been studied. So because we know that spatial memory depends on the hippocampus, this provides us then an opportunity to design experiments to discover what brain changes lead to memory changes during normal aging. Let's go on to the second myth that I want to discuss with you. And this is the fact that no one now believes that major brain cell loss occurs during the aging process, during the normative aging process, as it does in dementing diseases. So it's important to be reminded that the major cell type in your brain, your neurons, are born largely before our birth and die with us at the end of our lives. So you have these cells, these neurons, essentially your entire life. They do not divide or re-enter the cell cycle like other body cells, for example, skin or liver cells. Now, this is what was thought about brain aging when I became interested in the aging process, memory and aging in the early 1970s, where on the left are examples of these are brain neurons, age 20, and by the time you get to age 90, they look rather shriveled and horrible. This was a picture when I started to think about aging brain and memory, and we know now that this is completely wrong, and I can give you reasons for why these experiments happened and what went wrong with them, but this is wrong. There is no major cell loss with aging in the absence of dementing illness. Yes, you lose a few cells, but it's very, very small numbers. So, for example, when hippocampal cells, these are real cells that are filled with fluorescent dye, and when they're counted across the hippocampus and the three main cell types in the hippocampus, there's no cell loss, not in rats, mice, monkeys, humans, across our lifespan, in this major structure that is important for memory. So, more good news for those of us who are at least hoping to age successfully is that many cell functional characteristics are also preserved in the aged brain. In animals, it's possible to examine the function of live cells, and my lab has found that the properties of old hippocampal cells are similar in most respects to younger animals. The old cells are actually very healthy. So you might be sitting there saying, okay, there's no cell loss. What is normal brain aging? If, at least in the hippocampus, there's no cell loss. The cells that remain are healthy. Why do we have memory changes as we grow older? Well, we do know that two important changes occur in the brain: reduced numbers of connections and reduced plasticity. Okay, so I'll start with a change in the number of connections between brain cells and a loss of synapses first. We know that this, the brains of humans have 86 billion neurons, and each of those cells has about 10,000 synaptic connections. This is an amazingly interconnected structure. I'll start with the connections between cells. The axons of these neurons project to transmit information from one cell to another. This is the axon that makes a contact with the next cell. And most communication in the brain occurs by one cell sending electrical impulses down the axon to the other cell and to the end of this cell. And at this gap, the synapse releases chemicals that crosses the gap and then causes an electrical disturbance in the cell to which the other neuron is projecting. So this cell is now active. This cell then becomes active after. So while you do not lose hippocampal cells, during aging, older humans and other animals do lose these synaptic connections. Okay, so this is critical. And I just told you also that another thing that changes is synaptic plasticity. What is synaptic plasticity? Well, this involves the ability of one cell to communicate with another through these synaptic connections, these synapses. And when a synapse is stronger, it produces a larger, a greater electrical response in the cell that it's connected to. It's believed that memories, then, are stored as increases in synaptic strength of connections between cells. And although actually in the aged brain, synaptic strengthening continues to occur quite effectively, the strengthening is not as durable at older synapses as younger synapses. And I've been able to show that how durable this strengthening is of these synapses is correlated with how good your memory is actually at any age. And this is important to remember then. So on point number 2, neuron loss is not widespread in aging, but there is reduced connectivity and reduced plasticity that occurs in the nervous system. So let's go on to my third point. And that is that no one now believes that the brain just sits back and deteriorates. So there really has been a fundamental shift, I think, in the thinking in the last couple of decades about brain aging, in that we now believe that the brain does not passively deteriorate. Rather, in successful aging, the brain is continually adapting in ways that suggest actually outright compensation in some cases. So what I'm going to do is to give you 2 examples, not to overload you. I'm going to give you one animal example, one human example of this kind of adaptation to illustrate this. So in the animal experiment, this is one that I conducted that started with the following question and observation. We know that some hippocampal neurons from older rats have lost up to 1/3 of their synapses, okay? And the neuron, we know, also requires a certain level of incoming activity to become active and to transmit those electrical signals to the next cell, like I just showed you in a couple of slides previously. So in the face of this reduced input, how do these cells manage to fire? How do they become active and transmit information to the next cells as they need to? And we were able to show that the way that the hippocampus solves this reduced input problem is to make the individual synapses that remain actually stronger or more powerful. So fewer synapses can discharge that neuron. And I think this is evidence of biological compensation during aging at a single synapse level. Now, let me give you an example at the network level. And the observation is that retrieval can be very good in older individuals of salient past experiences, say last week or last year or 10 years ago. And we know that the hippocampus is involved in this, what's called autobiographical memory, or memories about yourself in the past. And so what I'm going to do is describe an experiment that suggests that older adults can recruit additional circuits to achieve accurate recall. And this is study is from McGuire and Frith. And they designed a study to look at brain activity during retrieval of old memories while people were scanned in the MRI, in an MRI scanner. And when the participants in the experiment were asked to retrieve these memories about their past life in the scanner, you see that on the left, the younger individuals, the left hippocampus becomes activated. When the older individuals retrieved their old memories, they used both left and right hippocampi to retrieve these experiences. So the actual neural circuits recruited to produce equivalent performance in young and aged individuals can be different. And this may potentially reflect adaptive compensation in the aged brain. So in summary of my third point is that the brain can adapt effectively during aging, suggesting actually great resilience and adaptability as we grow older. Now a fourth point. No one now believes that there is only one or a handful of genetic risk factors that protect or accelerate brain age-related changes in cognition. There are many, and this has been a revolution in genetic science with the discoveries that we've made over the past years. Now, what I'm going to do is just give you a couple of examples. I could go on and on, but I won't. For example, having a particular variant of this KIBRA gene will make it more likely that you have good memory your entire life. But having the wrong allelic gene variant will mean that you will have a higher risk of contracting Alzheimer's disease. And even at younger ages, people with a particular variant of this gene tend to have poor memories. Now, another interesting gene, HDL, genetic variants that produce high HDL levels, for example, predispose people not only to have strong cognition, but have very, very long lives. So centenarians are people that are 100 years old or older. For example, centenarians, their siblings, their offspring, all tend to have high HDL levels. So these things actually really matter. So the summary of my fourth point is that while I've only given you a few examples of genetic factors that influence memory, many complex genetic factors work together to determine your cognitive aging trajectory. And for the fifth point, no one now believes that there's only one or a few lifestyle factors that protect or accelerate age-related changes in cognition. There are many, and I'm going to go through a number of these with you: exercise, metabolic syndrome factors, inflammation, diet, stress, social interactions here, just to give you a flavor. But there are many, many more. So let's start with exercise. Exercise, of course, is good for maintaining bones and muscle mass. What's the evidence that exercise is good for brain aging? We think it should be, but what's the evidence? So I'll give you one example from Gene Alexander's laboratory. He's at the University of Arizona and a colleague. Gene and his colleagues analyzed whether there's a relationship between the level of physical fitness and markers of a healthy brain. What he did was to use magnetic resonance imaging of 123 people between the ages of 50 and 89 years, and then they determined the aerobic fitness level score for each individual by performing treadmill tests. And what they found was that the better your fitness level, the more fit your brain appears to be. They found that a relationship between the level of fitness and brain aging, so that you have a higher fitness index, you have less of the brain aging pattern. So if you weren't already convinced and need a good reason to take your walks every day in the morning or in the evening, I think this is pretty good evidence that you should. As well as exercise, a good diet that helps control your body mass index clearly has important effects on the brain. And these data are from Lee Ryan's laboratory from the University of Arizona as well. And as body mass index increases, you see increases in loss of volume of the brain and also lower memory scores. And as a side, these effects are accentuated if you have uncontrolled hypertension. So it's good to keep your body mass index at a healthy level. It's also clear that as we age, we need to be concerned about avoiding brain inflammation. Wear your helmets when you are cycling or you're skiing, because head injury is still one of the best predictors of who is going to get Alzheimer's disease, especially if that injury is after age 50. So in an interesting study, again, this is from my colleague Lee Ryan's laboratory at the University of Arizona, suggests that control of brain inflammation is really important for optimal brain aging. Lee and her colleagues had two groups of people aged 52 to 92 years. These groups were education, age and education matched controls, all living independently. And the two groups, though, were based or formed on the basis of whether or not the participants had or had not used non-steroidal anti-inflammatories. So the controls did not have a history of using these NSAIDs, or non-steroidal anti-inflammatories of various sorts, and the NSAID group had a mean drug use of 10 years or more, a minimum of 4 years. Now, within this age range in both groups, in this control group, the non-users, red indicates brain volume loss, and you can just see the difference by eye between these two populations. There was a significant decrease, volume loss with age, in the very brain areas we know play important roles in memory. Over a similar age range, the NSAID user group showed dramatically less brain volume change as measured by MRI, suggesting that there may be a protective effect of anti-inflammatory drugs on age-related brain volume. Okay. It's also becoming clear that compounds of foods that are found in a Mediterranean or other forms of diets are clearly good for brain health. They have powerful anti-inflammatory properties. As it turns out, foods such as leafy vegetables, nuts, dark chocolate, and drinks such as tea, coffee, and thank goodness wine even, all have anti-inflammatory properties. And this is because they belong to a class of antioxidants, the polyphenols, that are contained in these plants. In particular, the flavonoids have these anti-inflammatory properties, and the flavonols are the predominant class of anti-inflammatories found in dark chocolate and in nuts. In fact, newer studies have found correlations between certain kinds of memory and ingesting a certain amount of nuts, dark chocolate, olive oil, and coffee. Usually I bring with me nuts and dark chocolate to pass around for people who listen to this and have no guilt in having these things. So the good news is that just by including these things in your diet, you're undoubtedly helping to reduce brain inflammation and may be boosting synapse health. Now, the effects of stress on health and behavior is becoming better understood. For example, it's possible to measure cortisol, which is the body's stress hormone, in people who undergo MRI brain imaging. So cortisol was measured, and when the volumes of people, when the cortical volumes were measured across different regions of the brain, the people with the highest cortisol levels also had the greatest shrinkage of the prefrontal cortex, or the frontal cortical regions. Now, this brain region is known to be very important for the kind of memory, working memory, a type of memory that shows some of the earliest changes during aging. I think this is a really interesting study, so I'm going to show you. Tai Chi exercises have also been appreciated for some time for their stress-reducing properties. And one concrete benefit has been shown in this study, where one group of older adults were trained for 16 weeks on Tai Chi, and the other took a health education training course during that same interval before they were given a vaccine at this point for shingles. And the Tai Chi group showed increased cell-mediated immunity even before the vaccine, and then after boosted immunity after the vaccination compared to the control group, suggesting that Tai Chi may have beneficial effects both for the brain and for your immune system function, which of course the immune system and brain work very tightly together. So more is being learned now about importance of social cooperation and altruism in successful aging of individuals. And as we learn more and more about what structures in the brain are active during naturally rewarding situations, using MRI technologies, it appears that behaviors that reflect social cooperation and altruism also activate or exercise these very brain reward areas that are fundamental. And older adults who were studied over a long period of time, of more than a decade, and included in their activities, volunteerism and altruistic behaviors had several associated benefits compared to people who did not engage in these behaviors. And these include improved physical health, greater longevity, and increased life satisfaction. So to summarize, I think my point number 5, there are many, many lifestyle factors that can and do contribute to successful aging. And I think you can see just from listing these several things from before and now that we've come a long way in understanding how many of these variables go together to produce an aging brain. Certainly you don't have, Alzheimer's disease is not inevitable, you don't have major cell loss, and the brain is continually adapting, and many genetic and lifestyle factors can alter your trajectory towards positive cognitive outcome. So in conclusion, I think what I've shown you this afternoon is that your brain is remarkably adaptable throughout your life, and there are things that we can do to maintain brain health. And if there's one message that I hope you'll all take away this afternoon, it should be to actually keep those synapses of yours active, just as you have been doing in the past half hour sitting here listening to me. I don't think anybody fell asleep, so you are exercising your synapses. So I'd like to thank members of Congress for the continued support of interdisciplinary science, which is at the very heart of the type of neuroscience that I discussed today, and at the very heart also of the Brain Initiative, which I think Corey Barkman talked to you about at the beginning of this 2014 session. I also want to thank you for allowing me to share my work and my thoughts with you about brain aging. And what I want to end by saying is that what I'm sure about is that you'll continue to see a revolutionary advances from the field of neuroscience that will significantly improve and impact our health and quality of life going forward. Thanks again, and I'm happy to answer any questions that you may have. Back there?
There was an interesting 60 Minutes piece. There's a small town somewhere in California, and I forget the name of this place, but they took a survey of these people for the last 30 years and then kind of been following people into their 90s.
Yeah, that's Claudia Kawas's study.
Yeah, exactly. Yeah. And anyway, I was watching it, and one of the things that was interesting at the very end, they were talking about the brain, and they had been doing some slice autopsies of the brain. They found that some brains actually had kind of these microstrokes or mini strokes.
Yes,
some did it.
This produces,
and so I was just wondering if you could just talk about that, because I think they also said that just because you can have brain scans of people who have excellent memory that have a lot of these mini strokes and they're doing fine, and then you have people that have lots of these who aren't doing fine. And so I was just, if you can talk about that.
So the guess, and of course we don't know because we haven't done all the studies, but the guess is that even if you have a lot of these pathological markers, if your synapses are still intact, then you'll be able to function fine. So one part of individual variability may be how well your synapses remain connected through the life. So that's my explanation, and we're working really hard to understand what people have called cognitive reserve, those people who are able to function with incredibly high quality throughout life. And we think that one of these factors may be this promotion or lifelong synaptic connection ability. And this could have genetic, and it's going to have both genetic and environmental factors that play in and regulate this. Your genes are really important, though. There are pockets of people, centenarians, also have their children and so forth also live a long time, but they're incredible modifying factors. Even if you have bad genetic makeup, there's some of those people with the wrong form of the ApoE gene that are great at age 90. And so what we're trying to figure out is what other genetic influences come in to mitigate the typically negative effects of those. Yeah.
what is coming next, and you have to concentrate. So you get your brains to work.
Right.
And I think maybe origami helps, and then like Sudoku, so you have to use your brains.
Yes.
I don't know.
Yes, I agree that, part of the Tai Chi is, part of it's the meditative quality to reduce your stress, but also it's not like you're not doing anything. You have to concentrate strongly to be able to balance and to do all the movements correctly, and that is really important. Right, everything goes together. But I have to say that Sudoku, you brought that up, I didn't, but there really has been no proof that memory is better after you do crossword puzzles or Sudoku or anything. So you get more and more and more skillful with crossword puzzles and Sudoku, but that doesn't generalize across other things. Whereas I think Tai Chi may generalize for many reasons across a greater range of memories, across the kinds of memories that we have.
I wanted to ask about social behaviors and interaction with other people.
Right.
You knew that maybe some alcohol use in the brain that last hours may signal that you dated other people for last months.
Right.
Have there been studies about the aging of the brain and the correlation between heavy alcohol intake versus who don't drink or who drink socially?
So what's obvious is that too much alcohol, of course, is damaging. And however, is there amount? No. So it depends on your metabolism, all sorts of things. But what I can tell you is that it's much better to drink some alcohol than no alcohol. You live longer, and it's correlated with better memory, in fact. So, ethyl alcohol is a free radical scavenger, so it protects, it's an antioxidant, actually. So some of that is protective. Of course, too much, if you've damaged your liver and so forth, you can't remove toxins and so on. And so I'm sorry I can't give you a prescription, but it's different for different people, but you should at least have a glass of wine occasionally.
People say, I don't want to drink because, it kills the brain cells.
That's wrong. It's just like they said you lose 10,000 brain cells a day as you get older. That's wrong too. So, no. Whoops. Somebody else? Okay, back there.
So what I picked up is that in a healthy brain, you can all expect over time to lose memory. But over time, is that a surrogate for all brain functions? You often hear stories about how when people age, some cognitive skills improve. They get better with certain.
Absolutely. So vocabulary, for example, we continue to gain words, well into our 90s. So there's certain things that you can pick that you say that's better. There are certain deep structures in the brain that don't change at all, or it would be very bad, such as the things that control your respiration and fundamental things. So, yes, you do lose synapses in some of the higher cortical areas, but the brain is very well preserved down in the basic brainstem regions where you really need it. And one really interesting change, if I can just go on and on, so one interesting thing I think is that in fact there are some modifications of the brain in the amygdala, in the part of the brain that controls emotional properties. Actually, it's been demonstrated that the older you get, the more positive you get. That is, you tend to look at life, or you tend to emphasize the positive and de-emphasize the negative. And you can actually see in the brain with different MRI activations that the brain is processing positive things differently than negative things. So that's one good thing about being older. Younger people tend to be more negative and focus on the negative more, whereas older people don't. And there's a structural brain correlate to that. The brain changed, but I think that kind of adaptation actually I think is very positive. There and then there.
I have a question. Do you think I've noticed with myself that my short-term memory is not as good as it should be? So you think, and sometimes I worry because I'm like, maybe I'm gonna be 50 and I'm not gonna be able to remember anything when they tell me something. So is it related to brain aging or?
Yes. So the frontal lobe that I was telling you about that changes earlier than any other brain structure, well, it's that memory where you go down to the kitchen, you went down there for something, and you can't for the life of you remember what it was. So what happens is that, you get these intervening thoughts that blows your short-term memory away, right? Those are your frontal lobes, and it's really depressing, but your working memory starts to get bad in your 30s, compared to what it was, it peaks out in your 20s. So your working memory goes quite early. So this is aging. To me, it's all, it's development and aging. One back there?
Real quick, any insight into there's this myth about creativity, you get less creative over time. I realize that might be one step removed from the type of research you're doing, but can you say something about, is it really true you get less creative if, for example, they always say that mathematician,
right,
do your best work when they're,
there's always counterexamples of older individuals being incredibly creative. So to generalize, maybe for mathematics, although, I have to find a really good example of an older mathematician who did his best or her best work at that age. I don't have an example to give you, but I just can't answer that objectively. But
there's nothing to say you believe it or you don't believe it.
There's nothing to say that I can believe either way. I don't want to believe it because I see older people being very creative. Just like in 1970, I didn't want to believe that all older people are senile because I saw older people around me being fabulous. And so these are just feelings, and we need to study it directly there and then there.
There's some fairly new data about the impact of sleep on brain aging, and I wondered if you, you can think of that as a lifestyle factor or a behavior, but in some ways, sleep is, the quality of sleep often degrades as you age, so I was curious what you thought.
Yes, and there's also counterexamples. So Tim Monk has these 90-year-olds that sleep better than any 25-year-old you've ever seen. So there's always counterexamples. But Dick Bootson is a real expert on sleep, and is actually at the University of Arizona. And I just heard a talk from him recently. And our sleep tends to be a little bit more fragmented as we get older. And so it's harder to get the best quality of sleep because of that fragmentation. Now, that's a generalization. Not everybody is that way, but... So what he's done is that older people that get, say, 8.5, 9 hours of sleep, this should be just fine. What he's done is sleep deprivation on them. Force them to sleep an hour less. He's had fantastic luck. Their sleep becomes less fragmented when they do sleep. It tends to chunk together and so forth. So I think that there may be, not drugs, not anything, just restrict the amount of sleep you get and be really rigorous about it. And he's had fantastic results in this new trial that they're doing now, sleep study.
This is wonderful information that you just gave to us. One question I have is, do you have any information on the impact of the meditations on the brain?
Yes.
The kind of exercise people do to the brain.
So I also have another colleague at the University of Arizona, Al Kazniak, who studied this quite deeply. And the question was about meditation and the brain. And partly, it's hard to control for stress levels between people. New meditators, older, people who have meditated a longer time, because it tends to be the stress levels tend to be much lower in people who routinely meditate. So it's all caught up with the cortisol levels and anxiety, things like this that meditation really helps protect. So one correlation that's really strong is that it seems that regular meditators have less brain inflammation. So again, that's what we're all trying to achieve for a healthier brain synapses. Well, thanks for all the questions. This is great.